In our final video in our series on operons, we're going to look at how the tryptophan functions, which is the operon in bacteria that is used to regulate the production of one of the amino acids called tryptophan, which is the amino acid that we see here. So tryptophan has a relatively complicated structure as far as amino acids goes, and even just to make this single compound here, a total of 5 genes, which are what we simply call tryp-E all the way through tryp-A. So tryp-E, tryp-D, tryp-C, tryp-B, and tryp-A are all examples of genes that control the production of enzymes. And each of these enzymes is involved in the catalysis of one of the intermediates that eventually leads to the synthesis of tryptophan. So what that means is that a specific reactant is brought into the bacterium, and tryp-E catalyzes the formation of a different intermediate, and then tryp-D, another one, tryp-C, another one, tryp-B, another one, and tryp-A catalyzes the final reaction that will eventually result in the production of tryptophan. Now, none of this is really necessary. The point is that every single one of these proteins is needed to give this amino acid in its functional form, and the more activity that this operon has, the higher the concentration of tryptophan is going to be inside of the bacterium, naturally. The main difference in understanding how the tryptophan functions compared to how the lac operon functions is to look at the repressor. So normally we can see that the repressor is actually detached from the operator, which means that the default position of the tryptophan is on, because bacteria, for the most part, need to synthesize their own tryptophan rather than importing it through a transport protein, because tryptophan has a relatively large and cumbersome structure. Now, if the default position of the operon is on, we must need some way in order to shut the operon off, and we can see here that as the concentration of tryptophan inside of the bacterium increases, the more likely that tryptophan is to bind to an allosteric site located on the repressor, which, not surprisingly, is what we call the tryp repressor. So in this case, if we compare it to allolactose, which acts as an inducer, tryptophan actually acts as a compound that we call a co-repressor, meaning that tryptophan, when bound to the repressor, causes the repressor to activate and bind to the operator sequence, thereby preventing RNA polymerase from accessing all of the tryptophan genes and stopping the process of transcription before it begins. So if we can conclude about the general trends of tryptophan production regulation, as the concentration of tryptophan increases, the repressor will therefore become active into its active form and will therefore inhibit the production of tryptophan by blocking the RNA polymerase molecule from getting to the actual genes, and conversely, as the concentration of tryptophan decreases, this leads to the inactivation of the repressor because tryptophan is not able to bind to the allosteric site, therefore leading to the repressor falling off of the operator. And when the tryp repressor detaches from the operator, it causes the production of tryptophan to increase. In other words, as the more tryptophan is produced, the more tryptophan itself inhibits its natural production, leading us to conclude that this is a classic example of negative feedback, just simply done at the genetic level rather than at the level of hormone regulation. Now, If we were to stop here, that would be fine and good, and this is the primary method of inhibiting the production of tryptophan, but the trip repressor has a secondary regulatory mechanism which also functions in terms of negative feedback, and this is what we call the process of attenuation, which is relatively unique to bacteria for reasons that we'll get into when we look at how bacterial mRNA is synthesized. So if we look at the gene structure in between the operator and the actual genetic components of the trip operon, we find another regulatory sequence that we did not see in the lac operon, and this is what we call an attenuator sequence. And the attenuator sequence is characterized by a number of characteristic genetic sequences or base pair sequences. So if we look at sequence number 1 has many codons that code for the amino acid tryptophan, which has important methods of regulation that relate to the process of translation rather than transcription, whereas sequences 2 and 3 in the attenuator sequence are complementary to one another, as are sequences 3 and 4. And what this means, depending on which sequences are able to bind with each other, when these complementary sequences form base pairs, they result in unique structures which are what we call hairpin loops, and these hairpin loops have specific ramifications about how transcription is regulated. For example, here, if we have a high tryptophan level, that means that there is going to be a lot of tRNA that is bonded to the tryptophan amino acid, and if this concentration is high, that means that it is very likely for tRNA containing tryptophan to enter a ribosome, and for the ribosome to translate sequence number 1 relatively quickly without having to wait for this tRNA containing tryptophan to get in, which means that as the ribosome is reading this mRNA molecule, it's going to go straight through and block access to sequence number 2, which means here that a hairpin loop forms between sequences 3 and 4 in the attenuator, and when this happens, we can see that RNA polymerase was normally here, and this hairpin loop forming at this time causes RNA polymerase's hold over the DNA sequence that it is transcribing to weaken and causes RNA polymerase to actually fall off of the DNA molecule because of the tension that's created in the mRNA molecule that it is transcribing, which ultimately causes it to fall off and therefore causes the process of transcription to stop. Now, the reason why attenuator sequences are only relevant and found in bacteria is because normally you don't get transcription and translation occurring at the same time, because in eukaryotic cells, ribosomes are found outside of the nucleus, whereas RNA synthesis only takes place within the nucleus, meaning that this regulation method is not possible in eukaryotic cells. Now, while the high concentration of tryptophan leads to an inhibitory attenuator sequence, we can see that a different result happens if the concentration of tryptophan-containing tRNA is low. Because here, the ribosome has stopped at sequence number 1 because there are a lot of codons that signal for the addition of tryptophan, meaning that if the concentration of tRNA containing tryptophan is low, the ribosome is going to stall waiting for this tryptophan-containing tRNA, and as it is waiting, that means that the ribosome is not going to advance and not going to block sequence number 2, meaning that instead of getting a hairpin loop between 3 and 4, which causes the polymerase to detach, this hairpin loop is going to form between sequences 2 and 3 on the attenuator rather than 3 and 4, and as a result, the polymerase molecule is not disrupted from falling off of the DNA sequence, and transcription is going to be able to continue. And here, if this is the attenuator sequence, immediately after the attenuator sequence are the actual genes, meaning that RNA polymerase can now actually fully transcribe all of the genes necessary for the trip operon, thereby increasing the concentration of tRNA that contains tryptophan and leading to the negative feedback sequence. This concludes our series on both the genetic regulation of operons using repressors as well as our attenuator sequences. In the final video series of the molecular genetics unit, we're going to take a look and clarify a number of misunderstandings people have regarding mutations.